Good afternoon, everyone. This is Sherry Lynn Phelps, Agronomy and Program Seed Manager with Saskatchewan Pulse Growers. And today I have Andrea Laudner, our Communications Manager, who is also with Sask Pulse Growers. And she is the one that's been doing all the organizing and heavy lifting when it comes to these webinars. Welcome to the second webinar in the new Pulse series. We have 3 scheduled so far and look to add more as the year goes along. Webinars can be found on SPG website under News and Events tab. If you have suggestions or ideas for webinars, we would love to hear from you. Just before we get going on our webinar, just a little bit of information on those that have CCA or CCSC and how you can get credits, as well as the process that we're going to go through today. So to get your credits for CCA, you must be watching it live. For those who attended the webinar, Andrea will send out an email after the webinar requesting your CCA number. At this time, you can respond with providing her your number. If more than one person is watching from one computer, you will need someone to verify those in attendance and can include all the CCA numbers. Webinars will be recorded and posted to the SPG website for future viewing for those that are unable to attend or for those that want to have another look at it. Again, they will be posted in a similar place under the Communication tab on the Sask Pulse Growers website. So the process for today's webinar, we will mute all participants and we will take questions throughout the question box that's located in the GoToWebinar sidebar on the right. You can send questions at any time, but we will hold all the questions until the end of the section. For today's webinar, Dr. Beniza has divided her talk into 5 sections, and we can address questions after each section as well as at the end of the presentation. So let's get started. Today's speaker is Dr. Sabine Beniza from the Crop Development Center at the University of Saskatchewan. Dr. Beniza completed her undergraduate degree in Germany, followed by a master's in integrated pest and disease management and a PhD in plant pathology in the UK. In 1998, she saw the light and not only moved to Saskatoon, Saskatchewan, but also started working on pulse crop pathology in the Pulse Research Group at the Crop Development Center at the University of Saskatchewan. In addition to teaching and training of undergraduate and graduate students, she's developed a diverse research program comprising work on disease management strategies, resistance breeding activities, and more basic research on pathogen biology and host pathogen interactions. Her talk today is titled Managing Pulse Diseases in 2016, and it's going to cover various areas of pulse diseases and disease management. I will now turn over the controls to Dr. Beniza.
Good afternoon, everybody. I'm very happy that you all decided to join me for this webinar session. So for those among you who are experienced with pulse diseases, there is probably information in here that you have heard before, but a refresher never hurts. And for the newcomers to pulse disease management, I think there is quite a lot of information here. If you've heard me talking before, you know that I always start my presentations looking at sort of the weather and in particular the rain. And if I, oh, here we go. And in particular, I would like to well, look at precipitation. So in the past, I've shown these graphs before. And with the emergence of root rot, I actually look at precipitation, splitting up the rainfall patterns based on early and late season precipitation. So this is unfortunately in millimeter, whereas many of you probably are more used to inches, but as scientists, I guess we always deal with the metric scale. So I plotted here precipitation during the growing season from April till October from 2005 to 2015, and I split it into early season precipitation from April to June and then from July till October. So if you have a look at this, you will notice that between 2010 and 2015 we had a lot of early season precipitation. and in some of these years we also had a lot of late season precipitation. The average in Saskatchewan is sort of here about 350 millimeters per year. So obviously what is missing here is still the precipitation we get in the winter as snow, which I haven't added here. For the spring rain from April till June, the average is somewhere around 140 millimeters, and you can see here quite clearly that in 2010 and in particular 2012 and 2014, we had nearly double as much precipitation during those periods than we have had in previous years, and based on what the average usually is. And that is what triggered many of the root rot problems, and I will talk about that later on. 2015 fortunately was a bit drier in the spring, but you notice that sort of late season precipitation was quite high, and actually was the highest in the past 10 years. And that's why when you look at the cropland topsoil moisture conditions that are published by the government of Saskatchewan in their crop reports, you will see that on across the province topsoil moisture at the moment is still adequate. There are some pockets here where there is probably a bit of excess moisture, and there are sort of the first pockets showing up where things are getting dry. And the reason for that is, if you look at rainfall, cumulative rainfall from April till May, you will notice that overall we have had very little rain. Usually for this period we would have somewhere around 150 millimeters of rain by now, and that would be in these sort of darker orange colors here. So it's only really in the southwest where there has been enough rain so far, whereas in the rest of the province it has been very dry. But fortunately there is still quite a bit of reserve in the soil, and we have seen more showers coming through and thunderstorms, so I think things look quite good. However, obviously with the showers, that also means there is the potential now for diseases to get started, because almost all of the diseases we deal with in the pulses are triggered by rain and moisture canopy. Many of them need the rain in order to spread. Okay, so that sort of sets the stage, and now I want to move on to lentil diseases. I think with the good lentil prices we have seen a significant increase in lentil diseases. Rotations have been shortened. There may be new lentil growers who want to try it this year, so I think it's probably very good to have a look at the lentil diseases. The number one lentil disease we have in the province these days is anthracnose, and I have some pictures here that show some of the characteristic pictures. So this fungus causes beige brown lesions, which you can sort of see here. These are very early lesions, and you realize they are not very conspicuous, so you really have to look very closely to really see them. What you will also see once the disease has established is quite a bit of leaf drop, which you can see in the next picture here. And then you see these stem lesions developing, and these stem lesions can be quite dangerous because they basically, if they enlarge, can girdle the stem, and then the whole plant part above the stem lesion may die. And when this happens, you all of a sudden start seeing these dead patches in your lentil crop. Now, we do have some resistance to anthracnose, and I would like to refer you to the varieties of grain crops that is published every year. So here is sort of the top of the 2016 guide. Now, when it comes to resistance to anthracnose, it is important to know that there are two races that have been described. Race 1, which is the less aggressive race, and then race 0, which is more aggressive, and probably because of that has become the more common race. That's sort of unfortunate, because if you look in this varieties of grain crops guide under lentil, you will notice that there is only one column for anthracnose, and it is for race 1, so the race that nowadays is less common, whereas there is no column for race 0 resistance. So question obviously is, why only race 1? resistance in varieties, and the simple answer for that is, well, there is really no race zero resistance in the cultivated lentil germplasm, so we haven't found anything that really stands up to this aggressive race. However, we have found resistance in Lentil ovoides, which is a sort of a wild relative of the cultivated lentil. Unfortunately, Lentil ovoides has some characteristics that you wouldn't like as a lentil grower. So on the picture here to the right, you see that it grows in a quite prostate form, so in no way would you want to see this in a lentil variety. And if you look at the picture below, you will also notice that the seed is very, very small, and it's a red cotyledon species. So it takes quite a bit of breeding effort to when you cross it with the cultivated lentil to only retain the resistance to anthracnose and get rid of all the other characters that are undesirable. And that's why we haven't really gotten good cultivated lentil varieties with race zero resistance. But we are intensively working on this and hopefully have something in a short period of time. So the second disease I want to talk about is Ascochyda blight, and you notice that I sort of put a question mark here. Ascochyda blight used to be the lentil disease #1 when lentils started to be grown here in the province. But really, when we look back, the last serious outbreak of Ascochyda blight was in 2005. So that's quite a while ago, and I think what this is really the success story of the lentil breeding program. We have bred quite good resistance into almost all the lentil cultivars that are available now. And I think what has happened is that therefore, as a result of this, the population of this pathogen has just imploded and hasn't really been able to come back, which is great news for breeders and for growers. Now, if you look at the top picture there, you can see the lesions. So they are quite distinct in that they have this dark brown border, and you can see here, if you sort of look a little bit closer, that there are these little dots in the center, and these are the fruiting structures which you wouldn't see if you were looking at an anthracnose lesion. However, with Ascochyda blight, you also have leaf drop and stem lesions. Now, going back to the varieties with good resistance, to the crop guide here, you notice that if you looked at the Ascochyda blight column, as I indicated, almost all of our varieties have pretty good resistance to Ascochyda blight. A third disease I want to talk about, and you've maybe heard me talking about this before, is Staphylium blight. Well, again, it's sort of a disease that has light beige lesions that then eventually coalesce, and then sort of the whole leaf turns brown. And you also have leaf drop. And in the bottom picture, you sort of can see what can happen in a severe outbreak. And this is from Saskatchewan, but it's not very often that you see it at that severe of a level. Now, we have done a little bit of research on this. It's not easy to work with this disease because it's actually one of the diseases where the spores are airborne, and that makes it very difficult to do field research with it, where you want to control how much fungus you get into your plots. Anyway, based on our research, we have the impression that if you get early to mid-flower infection, then you may actually see some yield loss, seed staining, and seed infection. However, very often this disease comes in relatively late in the season, sort of the tail end of the growing season, where we think it probably doesn't cause much damage anymore, and if it's really late, it may actually work like a natural desiccant because it defoliates the leaves. Now, the next group of diseases I want to talk about are Sclerotinia white mold and Botrytis gray mold. So they are sort of these are two different organisms, but they are a little bit like sister and brother. They often show up together because they both sort of thrive in the same conditions. So Sclerotinia white mold, as the name implies, produces this cottony white growth on the plant surface, and you can also see it here on the seed, this white stuff. That's the mycelium, that's sort of the body of the fungus, whereas Petridis gray mold has more like a fuzzy gray growth on the surface of the plant material. So in contrast to the other diseases I was talking about, it's actually very easy even for a non-specialist to recognize these two diseases because they grow on the outside of the plants. And both of these pathogens can grow on stems, leaves, flowers, pods, and infect the seed. They usually show up late in the season because they really need a dense, moist or wet canopy to develop. And so in years when you have very moist growing conditions right from the beginning on, when the lentil plants become quite tall and lush, that's usually when you can run into problems with Sclerotinia white mold and Petridis gray mold. And in those conditions, very often lentil plants tend to lodge, and with the lodging then you get a sort of even denser mat of biomass, lentil biomass, and it doesn't dry up anymore, and that's when these two diseases really get going. Now, Sherilyn told me that she already received some sort of requests about some white spots on lentil. We have seen this quite often in the past years. This is not a fungal disease or a biotic, a microorganism that causes this. This is physiological, maybe because of a combination of sort of initially dry conditions and then moisture. What is likely happening is that cells basically burst, and that's why they look. You see these sort of lighter colored areas on the leaves. Lentil plants will usually grow out of that, so it's not something to worry about. Okay, so disease management of lentil. For the diseases like Anthracnose and Ascochyta blight, we strongly recommend a 4-year rotation for these diseases, because otherwise you run into the risk that the inoculum, the fungus, builds up in your fields, and then if you run into a year where conditions are conducive, you will just see earlier outbreak and much more severe outbreak, which is more difficult to control. I don't include Staphylium blight here because, as I said, Staphylium blight produces airborne spores, and that's the main way of how it is spread. It's a very common in the environment, so rotation probably doesn't have as much of a benefit here than with Anthracnose and Ascochyta blight. Obviously, if you have a choice, you want to use cultivars with resistance, and I already pointed out some of the limitations in terms of Anthracnose. What is really important is scouting, and you want to start scouting early before the plants sort of get into flowering, and definitely before your canopy starts to close. So the best time for fungicide application is somewhere around the 10-node stage. So obviously you want to know before that period whether you already have some disease in your canopy. I want to emphasize here that these diseases like Anthracnose, Ascochyta blight, but even Staphylium blight are driven by rain, because even if the spores are transported by wind, the spores still need leaf wetness in order to infect the plants. So we have had sort of in many areas showers and thunderstorms in the past few weeks, and so I think things are happening in the fields now, so it's probably a good time to really get out there and have a look whether something is showing up. Now, it's not a one-time exercise, and should be done on a regular basis, and in particular after rain. So I can't emphasize this enough. Rain is really the determining factor here whether disease will be initiated or not. Obviously, many growers have large fields, a lot of acreage. So what I recommend, or what we recommend, is really that you focus on the high-risk areas, and those are the areas that are the wettest, and that usually means where, for whatever reason, the canopy is the densest, doesn't dry up very quickly, where you see lodging or whatever else creates an environment that is more moist than in other parts of the field. What is also important to remember is that all of these diseases start from the bottom up, so it's not good enough to just sort of glance across the field and think, well, I don't see anything, everything is green. You really have to look down into the canopy, to the bottom of the plants to see whether disease is starting. Now there are decision support systems for fungicide applications available, and I have the one that is shown on the SAS Pulse Growers webpage, as well as that of the Ministry of Agriculture here, where you're sort of guided through the process to decide whether a fungicide application is necessary. So you will be asked to look at the plant stand, and obviously the denser the plant stand, the more likely of having conducive conditions in these fields, because you have more moisture retention, and that will enable these fungi to infect. The second, or the next factors are weather, so whether there has been rain, which would indicate, yes, you probably can count on some infection already having taken place, and then sort of looking forward, whether there is rain in the forecast, because that will then mean, okay, the next cycle can be initiated and more infection will happen in your field. So the 5-day weather forecast is probably the weakest link, because if you sort of pay attention to weather forecasts here in Saskatchewan, you will notice that they are not very predictable, not very reliable, and very often it's only in the morning where you know whether maybe there will be some rain in the afternoon. And then obviously the last component here under D is to determine whether you already have disease in your field, and clearly whether and how much disease you have will increase your risk for having more problems. And then you sort of count up the numbers, and it will then sort of tell you whether how high your risk is and whether a fungicide application is recommended. So this is the scenario for Ascochyta blight and Anthracnose, and Staphylium will be controlled through this way as well if there is a danger of early Staphylium blight infection. Now the story is quite different for Sclerotinia white mold and Petridis gray mold, because these diseases, as I already pointed out, mostly develop in thick stands after canopy closure. So that's sort of late July, early August into September, depending on how late harvest is. So if you have Sclerotinia white mold or Petridis gray mold problems, that usually means you have a really thick lentil stand. And that causes a serious problem in terms of fungicide application, because your fungicide will have, you won't have the ability to get your fungicide down into the bottom part of the canopy. So very likely you will really only reach the top third part of the canopy if you're lucky, but further down below, that's where basically these diseases can spread very rapidly. So for these two diseases, unfortunately, fungicide control is not a very good option. So if you have any questions at this point, it may be a good time to ask them before I move on to pea diseases.
Sabine, it's Sherry-Lynn here. We have a couple questions. The first one is it really important to be able to differentiate between the diseases such as Ascochyta and Anthracnose? In other words, do I need to know whether they are different or identify them in the field for different management practices?
In terms of disease management, it's probably not important, because as I indicated here, the management strategy is the same. So if you see lesions early on in your crop and conditions are conducive to infection, it doesn't matter whether it's an Anthracnose lesion or an Ascochyta blight lesion or a Staphylium blight lesion, you will follow the same disease management categories. It is, however, important to sort of understand that for Sclerotinia white mold and Petridis gray mold, it's a different story. But as I pointed out, it's much easier even for non-pathologists to recognize them because they grow on the plant surface, and you should be able to recognize that. You will never see Ascochyta or Anthracnose or Stemphylium grow on the plant surface.
Okay, thank you. The last question is, would Stemphylium blight cause significant yield loss?
We don't have hard data on that, so I can only speculate. And I think if you have an early infection, I think you can encounter yield loss, yes.
I lied, it's not the last question. There was one more. What is your recommendation for the best control for white and gray mold?
That's a really tricky question because quite honestly, we don't really have very good management options for those two diseases. The best option is manage your canopy, but that's obviously very tricky because when you seed, you don't know what the growing season will look like. But basically, if you knew that it is going to be a wet year, you would probably try to seed at a slightly lower rate, just to make sure the canopy is a little bit more open. That's probably your best option.
Okay, thank you. You can carry on to the next section.
Okay, so moving on to pea diseases, that's relatively straightforward because the only really important pea disease we are dealing with these days is Ascochyta blight or Maxrella blight. And I think all pea growers and people working with pea crops will be familiar with those. So what you will see early in the season are these, or early on, are these purple-brown lesions that increase in size. So the top picture here I think shows quite nicely these very early lesions, and then the bottom picture shows what it looks like if you sort of have moist conditions, rain, etc., what it looks later on. So the lesions eventually enlarge, they coalesce, and then the whole leaves turn brown. Ascochyta blight of pea will also infect stems and flowers and pods. Now, for various reasons, really your best management strategy with Ascochyta blight of pea is a good rotation. So giving enough time, and we're talking about a 4-year rotation as well, for whatever fungal biomass is in your field to deteriorate so that you don't have that fungal biomass in the field when you grow the next pea crop. The reason why I say this is probably your best management strategy is because with fungicides, the efficacy is very variable. That has been shown in many field trials in various parts of the province and by various research groups. In some years you get good control and that translates in yield loss. In other years you get some control but it doesn't translate in yield loss. Sometimes you get no control but you see some yield increase. So you can't predict, it appears, whether a fungicide application really translates into a yield gain. So it is maybe more of an economic decision in peas whether you decide to spray a fungicide or not for that very reason. A fungicide decision support system very similar to what I sort of walked you through for the lentil has also been developed for pea. This was done in Alberta, and they find that quite useful. So that may be a way of sort of helping to make that decision, but you probably hear that I am a little bit skeptical when it comes to fungicide application in pea. Any questions to pea diseases management?
We have one right now, and the first, is there any truth to the growth promotion claim in some products like Headline?
So we looked at this over the last 3 years. Now, admittedly, those were well, I guess they were relatively wet years, and we found very little evidence for that. Now I pointed out that we had wet years when we were doing this research, because if you look into the literature, one of the benefits that products like Headline give to the plant is that water in a better way, so they may have benefit under dry conditions, and obviously we didn't look at that. So at least under wet conditions, I would think no. And as a plant pathologist, I want to emphasize that Headline is a very good fungicide, and if you spray it as a growth promoter, you may cause problems with its function as a fungicide. So that's why I definitely don't like to see it used in situations where it isn't used as a fungicide. It's too valuable as a fungicide to risk it.
Do you have any information about the plant parasitic nematodes affecting pea crops?
I don't. And I mean, I think it's more of a quarantine issue rather than sort of a disease issue here, in that it sort of causes yield loss, at least so far.
Okay.
So it may be more of a trade barrier issue.
There's another question, kind of getting back to, I think, the Headline question. Any thoughts on the product called Radiate on peas and lentils, which is a growth hormone?
Well, I'm not a specialist on growth hormones, so no, I'm afraid I don't have any opinion on that. Sorry.
Okay. Thank you. We have one root rot question, but we'll hold that till the root rot section.
Okay, perfect. All right, so obviously fungicides are an important tool in the pulse crops, and I hope everybody sort of has the guide to crop protection that is published every year. So the message that I want to emphasize again and emphasize every time I talk is rotate fungicide groups. So different groups, different modes of actions. And just sort of to really make sure everybody understands what I'm talking about is I put together this table. It's probably not including each and every fungicide that is registered in the pulses, but obviously the list is quite long. When I came to Saskatchewan, I think we had one, which was Bravo. And yeah, it's sort of great to see much more choice. So I listed those. At the very top you see I listed the chemical groups, and then below the resistance risk, and you see that it ranges from low to high. And then just to make it really easy to see, for each of these fungicides I sort of highlighted what chemical group they belong to, and sort of you notice that it is really, the group is really dominated by group 11 and group 7. So what I want to emphasize here, it's really a question of rotating chemical groups. So just having different names doesn't mean you're doing a good job. So for example, if you pick Elatos and then you rotate it with Lance, Ag, or Priaxor, you're not really rotating chemical groups, right? So the message is your rotation should be very colorful based on this table. So that is really, really important. So why are we so concerned about it? And maybe sort of it really sinks in better if you understand in more detail what is really going on in the field. So when we talk about fungicide insensitivity or fungicide resistance, what that means is basically the fungus becomes insensitive or resistant to the fungicide, which means the fungicide doesn't work anymore. So there are a range of risk factors. Single mode of action is one. So that's, for example, why the strobilurins are in a high risk category, because it's a single gene that only has to change in the fungus, and then it is insensitive to strobilurins. The biology of the fungus is also important, how diverse it is, how often it can reproduce during the growing season, how big the population is. then airborne spores that can determine how quickly any insensitive mutant can spread. But then in the end, what is really is the frequency of application of a particular fungicide or a particular fungicide group. So, as I pointed out, if we take the example of a single mode of action fungicide, this requires only one mutation in a fungus. So here on the left, this green rectangle is supposed to be my field. The dots are fungal strains. The white ones are the ones that are sensitive, and the red one is a mutant. So it's not as if the fungicide spray will induce the fungus to mutate, but these mutations are random, and they just happen, and they happen through UV or cosmic rays or just cell replication errors, and that there are other reasons why you see mutation as well. And I mean, mutation, well, I guess we look at it in a negative way, but in a positive way, mutations is what drives evolution, and we would still be in the cave if we didn't have mutations, right? But anyway, so mutations happen randomly and naturally, so that's not the problem. But what is the problem is what we do with these mutations. Now, mutations are, well, happen maybe 1 in a million times or 1 in 10 million times. So when you see these numbers, you probably think like, well, that's a pretty rare event, so what's the issue here? Well, it's maybe relatively rare if you think of human populations, but if you think of microbial populations, 1 in a million and 1 in 10 million is not very rare. And just to sort of emphasize this fact, I found an example in the literature which is not related to pulses, but to powdery mildew of barley. So someone sort of went through the exercise and actually estimated how much fungal reproduction you have. So they estimated that one lesion, and you see a picture here of a powdery mildew lesion on a barley leaf, produces about 10,000 spores per day. Quite a lot. Now, if you assume that 10% of a leaf area is infected, then you have about 100,000 lesions per square meter, or 1 billion spores per square meter or a day, or 10 trillion or 10 to the 13 spores per hectare and per day. So I think that sort of brings the message home that microbial populations are just gigantic compared to sort of those of higher organisms. And if you now go back to even the rarer mutation rate of 1 in 10 million individuals, that still means that you have 10 million or 10 to the 7 mutated individuals in the population per hectare and day. Now that sounds very scary, but obviously not each of these mutated individuals survive. Many don't ever land on a leaf and can cause a mutation, so may have no impact on the population. But I think this really shows that when it comes to microorganisms, it's a completely different ballgame. So what happens now with this mutant? So I've shown you that mutations are relatively common in microbial organisms like your plant pathogens. So going back to this field where we had this one individual that had mutated and is now insensitive to your single mode of action fungicide. So you spray, you kill all the white ones that are sensitive, and the red ones increase. Maybe some insensitive ones, sensitive ones fly in, you spray again, kill those, but the red ones survive and increase. So this, if you repeat this over and over again, you're basically selecting for the insensitive individuals, and they then take over the population, and eventually you end up with a field where you only have your mutated fungicide resistant strains in the field. And that then means, okay, at this point your fungicide is useless. So that's why it's really important to rotate fungicides, in particular those that are at high risk for this scenario to develop. All right, any questions to fungicide insensitivity?
Yes, there's one question on that, and that is, what would be an optimum sequence for fungicides if planning for 2 applications?
Well, I would always recommend for the second application to be either multi-mode of action with low risk or at least a combination product. Because if you do that, then you should sort of kill everybody in the field, every strain in the field. Whereas if you just took, let's say, a pure strobilurin project at the end of the season, you may still sort of favor those individuals, so those strains in the field that are insensitive to the strobilurin, but would be killed with a combination product or a multi-mode of action product.
Thank you, Sabine.
Okay, so moving on from fungicides to root rots. So I showed you sort of at the very beginning the rain patterns over the last 10 years and pointed out the really wet years from 2010 to 14, where sort of root rots really came to the forefront of our pulse production problems here. So I also showed you this cropland topsoil moisture condition map, and well, I mean, it's adequate, it's nice right now, and what we really want to hope is that, okay, we get enough rain to sort of keep the crops growing, but we don't want these surplus patches there to expand, because that could very easily mean we will see more root rot again this year. Okay, so talking a little bit about root rots, there are several species that can cause root rots. There are Fusariums, Aphanomyces, Rhizoctonia, and Pythium. Now, common feature of all of these 4 organisms is that they live in the soil, infect roots, they destroy root tissue, they often impact nodulation, so the plants are nitrogen starved, and then they prevent water uptake and nutrient transport. So really the plants get a double whammy here. They often occur in a complex, so it's usually not just one organism that you can find on these roots, and it's not very easy to determine which one came first and which one came second, and it may not really be important anyway. If you go into the field and try to identify the species, that's really, really difficult and usually has to really be done in the lab, but even there it can be quite tricky to determine what pathogens there are, because they compete with each other and some grow quicker and others grow slower. Now, we have been particularly concerned with Aphanomyces because it's quite aggressive, and the disease management options for Aphanomyces root rot are very, very limited. You've seen this picture probably, just showing on the left-hand side infected roots under controlled conditions, so sort of very likely not what you would see in the field, because this is a very classical symptom with the caramel brown discoloration that later on would turn black compared to the cream-colored roots on the right. So Aphanomyces is not just a pea pathogen, but it can also infect lentil, alfalfa, some alfalfa varieties, and some dry bean varieties. Chickpea, fava bean, and soybean have good partial resistance. So in areas where Aphanomyces has infested fields, they may be a good alternative pulse crop instead of pea or lentil. Now, it's the pathogen is favored by water-saturated soils, and that sort of explains why we have been seeing it, because we had these really, really wet springs. Now, Aphanomyces can infect the roots at any time of the growing season. So it's not as if when spring has gone, you can sort of release a sigh of relief here. but it's most devastating early in the season at the seedling stage, simply because the plants are just much more vulnerable. They have very little root system at this point. Nodulation is initiated. So if things go wrong there, the impact on the plant is much more devastating. So many of you probably have seen pictures like this over the last few years. These large yellow areas, often following water flows and slough edges, so sort of areas where there is a lot of moisture in the soil. So managing root rots is quite tricky, and really the most important advice is prevention, prevention, prevention. So crop rotation can be a very powerful tool, in particular for specialized pathogens like Aphanomyces, and Fusarium oxysporum is another one which is quite specialized. So in particular for Aphanomyces, crop rotation is probably the most powerful tool you have. And unfortunately that means really long rotations. We are talking about 6 to 8 years. Field selection, if you have that luxury, because we know that compaction promotes Aphanomyces. Heavier soils where you have higher water retention, more soil moisture promote Aphanomyces. So if you have a choice, that can be useful as well. Seed treatments are available as well. There are several actives against Fusarium and Rhizoctonia. Metalaxyl against Pythium, and many of the seed treatments that you can buy these days are a combination that includes metalaxyl, so that Fusarium, Rhizoctonia and Pythium are controlled. And then since last year, Aphanomyces has had, ethaboxam has had emergency registration for control of Aphanomyces or suppression of Aphanomyces on roots. Currently there are no post-seeding management tools available. In the Pacific Northwest they have done quite a bit of trialing with Fostrol and against Aphanomyces root rot, and have seen some sort of post-emergence benefits of Fostrol. I know that the company is trying to get registration here as well, and I talked to them and they said maybe in 2017 that will be an option here as well. Okay, any questions to root rots?
I think we could almost do a whole webinar on root rots. Lots of questions. So you mentioned some control options for root rots. Any particular experience with the one for Aphanomyces?
No, I know that there are some large trials going on, independent trials, but because last year the spring was so dry, I don't think the principal investigator really wants to talk about it at this point, because of the very dry conditions in spring, which meant there was really not much Aphanomyces root rot at that stage. And obviously a seed treatment will primarily protect the seedling in the early stages, right? And so if it's very dry, you probably don't see the true potential of any seed treatment.
Okay. Next question is regarding around the control options for Aphanomyces and the impact of tillage, as well as the application of products such as trifluralin that has been suggested to benefit control.
Okay. So, I mean, I pointed out the options that there are. So I think crop rotation is still, away from a susceptible host, is still the most beneficial management tool we have at the moment. Seed treatment will help as well, but it's not a silver bullet. So if you have a field that is infested with Aphanomyces, heavily infested with Aphanomyces, I don't think the seed treatment will do the trick, and you can expect to get a really good crop of this field. In terms of the herbicide, I actually have a slide here, and I quickly want to show this. So this is sort of some research that was done in the 1977, or published in 1977, under controlled conditions. So they tested two varieties. So these numbers, the varieties, 3 locations, and then they had the 0 is the control, the black, oh sorry, the black is the trifluralin, and the grayish one here is dinitramine, another herbicide. So you see that there is sort of a little bit of a benefit, but again, it's not sort of a silver bullet. So I think what is quite clear with Aphanomyces root rot, it's not as if we have one thing that will solve the whole problem. It will probably be a combination of seed treatment, rotation, moisture management in your field, and that brings me to the question about tillage. I can't say tillage is good or bad. It is all about managing any problems with waterlogging and drainage in your field. And if tillage solves that problem or improves that problems, then tillage will be beneficial. But that will be very soil and field specific. So you would have to really talk to someone who knows your field and understands what the effect of any mechanical measurement is on water movement in the soil.
Thank you. You can continue on now.
Okay, so because of the root rot problems and Aphanomyces problems across the prairies, I'm trying to talk a little bit about faba bean and chickpeas as well, because they are good alternatives in different areas of the province. So faba bean probably more in the somewhat moister areas, and chickpea in the southwest of the province. So with faba bean, there are diseases as well: ascochyda blight, chocolate spot, leaf rust, alternaria, and sclerotinia, of which probably the first 2 will be or are our main concern. So ascochyda blight, it's a distinct pathogen from lentil, pea, and chickpea. Each of these crop species has their own special species of ascochyda blight. But obviously they look very, very similar here. You have the grayish lesion again with the characteristic dark outer margin. And again, if you sort of look closer, you will see sort of a peppered spots in the center of these lesions. And these are the fruiting structures, and that's sort of a diagnostic for ascochyda blight. But diagnosing some of these disease symptoms is really tricky, and we have experienced this last year as well, because sometimes you may mix disease symptoms up with herbicide damage. And I sort of show you some pictures here. On the left-hand side, that is a picture of ascochyda blight from France, and on the right-hand side, a group for herbicide damage from Australia. And if you look at this, you think like, wow, that looks very similar. So in situations like this, it's very important to sort of really ask questions or think back. As with the other ascochyda blights, this is a rain-driven disease. So if you stand in a field on the right-hand side there and it hasn't rained for 2 or 3 or 4 weeks, then it's unlikely that this is ascochyda blight. And then probably you should ask, well, have you applied a fungicide, because that is then the more likely reason for those lesions on the leaf. Chocolate spot is the other disease, and maybe the more prominent disease in the province. It's referred to as Petridis gray mold in the other crops, although faba bean has its own special species here that is a little bit specialized. So initially you sort of see tiny reddish lesions, and to get my cursor here, there you are, sort of these little dots. That's the beginning here of Petridis infection, and they then sort of enlarge, they coalesce, and then you eventually end up with the entire leaf turning blackish brown. So it can be very devastating when conditions are wet, when there is a lot of rain, and can develop very quickly. It ends up with defoliation, and then even if the stems are severely affected, with lodging of whatever is left. Again, with chocolate spot, can be under certain conditions mixed up with herbicide damage, with group 5 herbicide damage. And we actually saw this here on our research farm last year. So again, it's sort of important then to put these disease or these symptoms on the plant in context of weather patterns. If the weather wasn't really conducive for disease, it's likely another reason. Alternatively, what you will have to do is send these samples into a lab where they can try to culture any microorganisms out of this brown tissue. Leaf rust is probably a bit rare, easy to recognize because you have these orange-brown blister-like pustules on the upper and lower leaf surface. I don't think it will be a regular disease we will see in our fava beans. Alternaria is maybe a little bit more common than the rust. So you have these dark brown spots, and you can see characteristically you have these concentric rings here, which you wouldn't see with with chocolate spot or ascochyla blight. It's often secondary infection, so once you have Petridis or ascochyla blight, that's when maybe Alternaria comes in. So not a really very important pathogen on its own. In really wet years, Sclerotinia white mold can also infect fava beans. So again, you would see this cottony white mycelium and maybe black sclerotial bodies on the plant surface. So it should be really easy to recognize for anybody. But as I want to emphasize here as well, it will probably only show up under really wet conditions. So coming to management of foliar diseases in fava bean, obviously as with lentil pea and chickpea, crop rotation is important to manage the fungal residue. And then fungicides application will be an important tool. The recommendation right now is applications at flowering if you see symptoms in the lower part of the canopy and if conditions are conducive. So if you are in a period where it has been raining and there is more rain in the forecast. What is important to remember with fava bean is that the flowering nodes vary in their level of fertility. So fava bean, when they grow, will have 6 to 7 vegetative nodes, and then the next nodes will be flowering nodes. So the first nodes sort of will slowly increase in fertility, and if you've seen fava beans, you will know that they produce these clusters of flowers. So the number of flowers in these clusters will increase up to the 4th node, and then as the plant grows further, they will sort of decline again. The number will decline. So what this means is that your largest yield potential is sort of in the middle part of the fava bean plant, and that's obviously what you want to protect. So follow-up applications may be required as the plant grows, depending on the weather conditions. I may want to mention one more thing as well. In contrast to the other pulse crops, fava bean set more pods if they are pollinated, so they really benefit from bees. And obviously if bees or bumblebees or solitary bees don't fly for whatever reason, that may impact your fava bean yield as well. There is more information on fava bean disease management on the SaskPulse website, so I recommend that you have a look there. There is also quite some good information from New South Wales in Australia. They have been growing fava bean for many, many years, but do remember that sort of the growing conditions are different in that it is a winter crop there that starts off in the wet, often wet Australian winter, and the crops mature in the dry Australian summer with a longer growing season. So that's quite different. So the disease management recommendations obviously will be somewhat different there for that very reason. But if you want to learn more about the diseases as such, I can recommend that site. Very quickly about chickpea, the main disease is ascochyda blight here. So again, these beige lesions with a dark margin. Among all the pulse crops, I would say ascochyda blight in chickpea is the most aggressive. So it spreads very, very fast, and you really have to be on top of managing that disease. So it requires very close watch and probably some experience to really do a good job. There is a disease, a fungicide application guide available as well, so I show that on the right, very similar to what I showed you for lentil and pea, and so I recommend that you have a look at this. Even if you don't use it, it sort of gives you an idea how to approach ascochyda blight management and fungicide use in chickpea. Okay, so any questions? That's really all the information I have here today, but I welcome any further questions on particular issues.
Okay, so we have a few questions and kind of goes all over the place, but I'll start with the faba bean and chickpea one. What is the best timing for fungicide application for chickpeas?
Well, with the chickpea, because it is such an aggressive disease, the recommendation is that you actually apply sort of per calendar seedling spray. With this disease, you can't really afford to start managing it once it has established in the field, so you have this preventative spray very early in the season, and then any consecutive spray will depend on whether there is disease showing up in the field and weather conditions. So after that preventative first spray, then you would use something like this disease, the fungicide application guide, to decide whether additional sprays are necessary.
Thank you. Just a reminder for the audience that if you have any questions, please just type them in that little box on the right-hand side, and we will answer them in order that they come in. Another question, going back to the white speckling in lentils that was not considered a disease, and a question revolving around why the cell walls would be breaking. What is the cause for that?
I don't think anybody really knows exactly what is going on. So, I mean, we've tossed this question around here in the sort of research community, and the best answer we could come up with is that it may have to do with going through a period where there isn't a lot of water in the plant, and then all of a sudden it takes up a lot of water, and so that may just sort of result in a water imbalance in some cells that make them burst or do something to them so that they appear in a different color on the surface.
Okay. Next question is a comment regarding Aphanomyces, and wondering why a 4-year rotation would not be enough for managing Aphanomyces when you're currently using a 4-year rotation such as wheat, pea, wheat, canola.
The reason is that in contrast to these fungi that cause your typical leaf diseases, this pathogen Aphanomyces produces spores that are very, very resilient, so they can survive for, well, at least a decade in the soil, maybe even longer. Whereas with the other fungi, they will die in a period of 3 to 4 years. So if you have an infested, an Aphanomyces infested field, and you grow a susceptible crop again in 4 years' time, there are still too many living spores of this pathogen in the soil. Whereas if you went back after 6 or 8 years, the number of surviving spores will be much, much lower, and so your risk is lower. of getting an infection and damage in your crop.
Okay. So if you aren't having issues or have identified a Phenomyces in your field, then the 4-year rotation should be fine. Is that correct?
Yes, that's correct.
Okay.
And that's why it's important to figure out whether a field is infested with Aphanomyces.
Okay. On peas and lentils still, what is the optimum timing for fungicide application when you're looking at 2 applications of fungicide versus just a single application? Is there different kind of timings that you should be doing if you're going to plan on doing 2 versus if you're just planning on doing 1?
So, I mean, obviously the first one would be based on whether you start seeing symptoms in your pea or lentil field, and you would really want to identify the very early lesions, right? I mean, if your plant is already half brown, that's sort of, you missed the train a little bit, and that's why regular scouting is so important. So your first application should be based on that, and that will be dependent on the weather. So with the rain we have been having in many parts of the province now, I would suspect that there are probably some symptoms showing up. So in such a situation, an application before canopy closure would be my recommendation. If it then continues to be wet, you would probably want to consider a second application in maybe 3 weeks or so, because the efficacy of a fungicide will decline within 2 to 3 weeks, plus you will see substantial amount of new growth that is unprotected.
Okay. The last question, unless another one comes in, is a comment regarding postponing seeding dates for peas by 7 to 10 days, increase their nitrogen applied at seeding in the hopes that it'll improve the chances against root rot. Any thoughts on that management practice?
Well, I think as sort of a standard approach, it may work or it may not work. I mean, obviously, whatever gets your pea seed germinating quickly, the plants developing very quickly, will be beneficial in the sense of that it may then be able to somewhat outgrow the pathogen. And if that means you achieve this by waiting a little bit longer because the soil is somewhat warmer and maybe you're lucky it's a little bit drier, then that will probably benefit. Nitrogen application, I mean, you obviously have to be a little bit careful, because if you apply too much nitrogen, you may then prevent from the nodules from forming, because there is sort of a bit of a competition between your fertilizer application and bacterial nodulation. But in general terms, obviously, if the plant is well nourished, again, it may be able to compete better and may have a better chance at outgrowing the pathogen. I think I quite wiggled my way around that, did I?
Sounded good. I think that was the last question. So thank you so much, Sabina, for your presentation and the depth of information that you provided. It was fantastic, and thanks to all those that are participating on the webinar today. Just a reminder to CCAs that an email will be sent out that you will need to reply to in order to get your CCA credits. You can also provide suggestions for future webinar topics at the same time. We really appreciate any feedback or suggestions if you are willing to provide those. Our next webinar is scheduled for August 17th, so we have a couple month break, and it's going to focus on grain handling and storage of pulses and featuring a guest speaker from PAMI, Joy Agnew. We will also have a demonstration site out at Ag in Motion this year for those of you that will be attending. SPG has contracted Tom Wolfe to do some sprayer demonstrations, so if you have spray questions, it's a great opportunity to come and get those questions answered from an expert. So again, thank you for participating and joining us on our webinar, and we look forward to your suggestions as we move forward. Thank you, and have a great day.